everyone, and welcome. Thank you for joining us today for our coastal state discussion on microplastics in Narragansett Bay, hotspots, pathways, and solutions. I'm Meredith Haas with Rhode Island Sea Grant, and we are a state federal university partnership through the National Oceanic and Atmospheric Administration based here at the University of Rhode Island. And we're one of several programs nationwide. Our work brings together scientists, students, communities, agencies, and industry partners to advance coastal resilience and the sustainable use of marine resources. And so the Coastal State Discussion Series highlights ongoing research that directly impacts Rhode Island communities and marine ecosystems. These conversations are designed to create a bridge between science and decision making, and giving researchers and students a platform to share their work and to give community members an opportunity to learn more about the science shaping our coast and to engage directly with the experts. So you can find more information about this series on Rhode Island Sea Grant's website, where you can find not only details about upcoming events, but also information about past events, where you can also find recordings on our YouTube station. So we will have today's session already up on the YouTube channel as well. So without further ado, let's turn our focus to today's discussion. As microplastics become more pervasive worldwide, they pose a risk to both environmental systems and our health. And so just like other coastal environments, Narragansett Bay is subjected to an influx of plastic material from our urban watersheds with potential impacts on marine species and human health. And our speakers today will share the latest research on where microplastics are found in Narragansett Bay, how they enter the system and move through the estuary, and what these pathways mean for protecting New England's largest estuary. So please join me in welcoming Dr. Sarah Davis, a recent PhD graduate in marine ecology at the University of Rhode Island and now postdoctoral research fellow. She will share her research on microplastic distribution and sources in the Bay's surface waters, focusing on how physical dynamics and watershed inputs influence concentrations across space and time. And she will be joined by Dr. Victoria Fulfer, a recent PhD graduate from the University of Rhode Island's Graduate School of Oceanography, and is now a microplastic scientist with Gyres Institute. And she will present her work investigating microplastics in the sediments of the bay, including findings that estimate more than 1,000 tons of microplastics are in the top 2 inches of the bay seafloor. So together, their work offers the first comprehensive picture of microplastic pathways in Narragansett Bay, from watershed to surface waters and to the seabed. So thank you for joining us today, and we are excited to hear more about your work.
Hello, everyone, and thank you so much for tuning in today. My name is Sarah Davis, and I'm currently a postdoctoral researcher in the College of the Environment and Life Sciences at the University of Rhode Island. And I'm looking forward to sharing our recent research on microplastics in Narragansett Bay surface waters with you. I'd like to start by acknowledging the huge team effort that went into this project, which was several years in the making and could not have been accomplished without the efforts of many collaborators. I'd also like to give a big thank you to Rhode Island Sea Grant for funding this research and for giving us the opportunity to share the process and results with you all today through the Coastal State Discussion Series. As we dive in, I think it's important to first define precisely what a microplastic is, or perhaps more accurately, what a microplastic can be. The term is truly an umbrella, and there are several characteristics that can vary from one particle to another, which affect their behavior in the environment, their impacts on organisms, and our approaches to studying them. So the first of these characteristics is size. The generally agreed upon upper size threshold for a microplastic is 5 millimeters, and the lower size threshold is 1 micrometer, or micron for short. This means that the largest microplastic can be 5,000 times larger than the smallest microplastic, which can have a huge impact on the way they move through the environment and what happens when organisms encounter them. The next important characteristic is shape. Some microplastics are engineered to be small, such as microbeads that might be found in cosmetics or cleaning products, and we call these primary microplastics. Others have broken off of large pieces of plastic, which typically present as fragments, films, and fibers from plastic textiles. These we refer to as secondary microplastics. Density is another important characteristic, especially in aquatic systems, as particles' density relative to water can affect where they tend to settle within the water column. And density is primarily driven by polymer type, which is the base material of the plastic itself. And the majority of plastics also contain additives, which give them some aspect of usefulness. And as you can imagine, all of these variable characteristics strongly affect each unique particle's impacts in the environment, and also our approaches to detecting, isolating, and analyzing them. So previous research has demonstrated that microplastics have detrimental effects on the environment. Microplastics are easily ingested, which can lead to digestive blockages, reduced feeding efficiency, issues of nutrient absorption, and it's also poorly understood how microplastics move through food webs. Microplastics can also act as vectors, meaning they can adsorb a layer of chemicals, including organic pollutants, which can magnify their negative effects. And they can also acquire a film of microorganisms, including potentially pathogenic viruses and bacteria, which can travel long distances and possibly new routes on their microplastic host. These and many other documented effects cause negative impacts on the organism and ecosystem levels and also ultimately impact human health. This brings me to the introduction of our recently published study, which investigated spatial and temporal variation of microplastics in one of our planet's most critical habitats, estuaries. Estuaries, as you may know, are partially enclosed bodies of water where freshwater rivers meet the ocean, and they're among some of the most productive ecosystems on Earth. And while there's been a wealth of research on other anthropogenic pollutants, the transport and fate of microplastics is still not very well understood. We do know from previous research that densely populated urban areas tend to be sources of microplastics into estuarine systems. However, how these microplastics move once they're in the estuary and whether they exhibit any trends in accumulation remains really understudied. Surface water movement in estuaries is highly dynamic, and it's likely that both short-term variables like tides and longer-term variables like seasonal precipitation, river volume, temperature, and wind play a major role in the movement of microplastics. And these phenomena have been preliminarily studied, but they require long-term, highly replicated sampling efforts. And in general, microplastics research tends not to replicate because of how labor-intensive the processing and analysis can be. Well, we are always up for a challenge at the University of Rhode Island, so we designed a field survey to assess longer-term seasonal and shorter-term tidal microplastic trends in New England's largest estuary, Narragansett Bay. So Narragansett Bay experiences a strong north to south urban population gradient. This gradient has previously been linked to other anthropogenic stress gradients in the bay and recently to microplastic concentrations in sediments, which was accomplished by Dr. Victoria Fulfer, my co-presenter today. Freshwater river inputs from the Blackstone, Patuxent, and Taunton rivers are also concentrated in the northern part of the estuary. The bay also has diurnal tides, which are responsible for the fastest observed water currents in the system and creates a weak counterclockwise flow of water. So for seasonal sampling, we identified 6 sites throughout the bay located close to, sorry about that. We identified 6 sites throughout the bay, spaced equally between the east and west passages and north and south. And then for tidal sampling, we identified 2 sites located close to the mouth of the bay in order to capture the effect of tidal mixing between the hypothesized more polluted estuarine water and less polluted open ocean water. So how did we accomplish all of this? Well, we used surface water trawling with a manta trawl net with a pore size of 330 microns and equipped with a flow meter to capture the total volume of water sampled. We trawled at our seasonal sites in winter, spring, and a combined summer-fall season. Two replicate samples were collected each season, and this was repeated for 2 years for a total of 72 unique trawls. For high frequency sampling, we trawled 10 times in the same location over the course of one full tidal cycle, and this was accomplished in 2 locations. Samples were taken from the field to the lab, where they were sieved, floated in a hypersaline solution, and suspected microplastics were hand selected under a microscope. We also captured high-resolution imagery for all of our particles to document their characteristics, and all suspected microplastics were melt tested, which is a way to rapidly eliminate non-plastic material from further analysis. And then a 20% subsample of all particles which passed the melt test were analyzed for polymer type using Raman spectroscopy. Contamination control is a critical part of microplastics research, as it is very easy to accidentally introduce additional microplastics to the workflow, which can be misidentified as originating from the sample being evaluated. So this study was subject to rigorous contamination control measures. These included things like control clothing, which is orange as seen here, field and laboratory blanks, the use of laminar flow hoods during all processing, and the use of pure water for washing and preparation of all reagents. So what did we identify? Well, first we evaluated the characteristics of about 3,000 particles recovered from seasonal sampling. Overall, microplastics tended to be low-density, colorful fragments made primarily of polypropylene and polyethylene. So this makes sense when we consider our sampling method, which was sampling surface water, and is therefore biased towards buoyant particles. Denser materials, including textile fibers, likely sink quickly out of the water column, and so are not reliably detected by this method. And we did see some statistically significant seasonal variation. Well, what about our concentrations? So our concentrations are presented as microplastic particles per cubic meter of seawater. And I'll first present these two figures, which show the average microplastic concentrations across sites and seasons. And sites, as we recall, are numbered 1 through 6 from north to south in the bay. We see that there is generally higher particle recovery in winter, and that the northern portion of the bay is generally more polluted. And while these are important results, these figures don't necessarily capture the breadth of variation that we observed throughout the entire sampling period. So to help visualize this, I'll break these results out by season and sampling year. Okay, so this is starting to capture the variation in microplastic concentrations that we observed throughout the 2 years of the study. At first glance, it might look a little chaotic. However, to make sense of this, we made connections to seasonal phenomena that might drive microplastic inputs and movement. First, we can look at river flow. So this panel shows the total daily volume of river flow from the Blackstone, Patuxent, and Taunton rivers, with each colored panel corresponding to season. Additionally, since we know that wind strongly influences surface water movement, we can also look at the average originating direction and speed of wind conditions within each season. And while this might seem complicated, there are some really interesting patterns emerging that I will walk us through. So in summer of year 1, we observed high river flow, which corresponded with high particle recovery and a strong north to south gradient of microplastics. So wind conditions during this period were typical for the time of year, which means they were blowing out of the southwest. And that means they were pushing surface waters towards the northern part of the bay. Similarly, we observed high river flow in winter of year 2, which also corresponded with high particle recovery, except in this instance, we saw more particles in the southern part of the bay. And wind conditions were also typical for this season, which means they were blowing out of the northwest and pushing surface waters south more rapidly. So these results altogether suggest that the freshwater rivers flowing through urban areas are likely accumulating microplastics, particularly during periods of increased precipitation, and they're depositing them into the bay. And then where these plastics then tend to accumulate is then mediated, at least in part, by wind. And I'd like to zoom in just briefly to one site, which is designated as Site 5 in our study design. So this site was located in the Southern West Passage, right around Dutch Island. And we had very high microplastic recovery at this site in particular during winter relative to other sites and seasons. And our assumption is that the high recovery at this site was likely influenced by a few different interacting factors. Not only do we have the increased flushing that's coming from high river flow and the wind conditions that are pushing water south, but the West Passage is also fairly shallow and narrow and experiences a strong, almost continual subtidal residual outflow of water. And all of these drivers together are possibly contributing to a bottleneck of floating debris, including microplastics that are almost getting stuck in this area as water is being flushed through. So now we've considered our longer term seasonal drivers of microplastics, but what about our short term variables? Our high frequency tidal sampling also yielded high variation. At our West Passage sampling site, we identified a strong relationship between tidal height and microplastic concentrations, with the highest recoveries identified near low tide. So these results suggest that during flood tide conditions, less polluted ocean water is being pulled into the West Passage, and it dilutes the surface water concentrations of microplastics in this area. Our East Passage site also exhibited high variation throughout the course of sampling, but there were less distinct tidal-related trends. However, within the course of just a few hours at either of these sites, sampling recoveries varied by over 1,000%. And this has significant implications for survey design and for the interpretation of previous study results that failed to account for the influence of tides. A small number of previous studies have reported tidal conditions during microplastic sampling in estuaries, and our results support some of their findings and conflict with others. So it's difficult to make direct comparisons, especially to other studies because they use inconsistent methodologies. But at a high level, this suggests that microplastic movement trends are likely unique to the specific hydrodynamics of each individual estuary. The final but very important part of this study was comparing our field-generated data with ocean circulation modeling simulations. So because microplastics field sampling can be so cost and labor intensive, more studies have recently been implementing hydrodynamic modeling to simulate the movement of microplastics within marine areas. However, most of these studies have no ground truth in field collected microplastics data, which prevents validation of the results. So to see how ocean circulation modeling compared to field sampling outcomes in Narragansett Bay, we utilized a high-resolution model of the bay called the Ocean State Ocean Model, which is made from the Regional Ocean Modeling System, or ROMS. We released virtual microplastics from 3 hypothesized source locations, which in this case were the mouths of the freshwater rivers. And we tracked these virtual microplastics movements during our 3 simulated seasons. So we found that the model reflected broad trends in particle movement, especially the increased flushing and broader distribution of microplastics observed in winter, and the reduced flushing and exacerbated north to south pollution gradient that we observed in summer. However, when we track individual particle movement through our seasonal sampling sites, we found that shorter model durations, in this case 3 days, were more reflective of our seasonal findings than longer model durations, such as 7 or 14 days. And this fine scale particle movement is essentially a snapshot of what's occurring within the bay at any given point in time. And it's challenging to replicate this with ocean circulation models alone because at present, they can't account for the variability in particle characteristics that can influence movement, such as size, shape, polymer type, degree of weathering, and rate of continued fragmentation. So overall, this study's results have important implications for pollution management in Narragansett Bay and for microplastics research in estuaries globally. Given the high variability in concentrations driven by tidal and seasonal patterns, future pollution management strategies would benefit from emphasizing the importance of adaptive monitoring programs that account for both temporal and spatial fluctuations in microplastics. Seasonal peaks in concentrations, particularly in the northern, more urbanized portion of the bay, suggest that targeted efforts to reduce urban runoff into freshwater rivers and to improve wastewater management during high input river flow periods could be effective in mitigating microplastic pollution. These study results also emphasize the importance of replication in microplastic sampling the patterns that we identified relating to river flow, wind conditions, and tidal variation would not have been identified without robust replication. And while Mantotrol sampling for microplastics is a well-established field survey technique, it does present some key limitations, primarily in that it only samples the top layer of the water column and is constrained to particles larger than 330 microns. Smaller particles likely represent a significant undetected quantity of microplastics and nanoplastics, and these should be an emphasis of future research. And our results also suggest that ocean circulation models can be used as a valuable tool alongside robust field sampling. However, at present, they cannot replace field sampling and field collected data, and we must continue investing in robust field survey campaigns. So as we look ahead to ongoing and future microplastics research in Rhode Island, I'm excited to share that the University of Rhode Island was recently awarded a grant from the National Science Foundation to support microplastics research throughout the state. This is a collaborative EPSCoR funded project with colleagues from Rhode Island College, Roger Williams University, and Brown University, as well as community-based organizations and local stakeholders. The project is called SimCoast, which stands for the Socio-Ecological Impacts of Microplastics in Coastal Ecosystems. So over the next 4 years, we will be undertaking cutting-edge research related to microplastics in Narragansett Bay and its watershed, not only on the sources, concentrations, and movement of plastics, but also their presence and impacts on local species of interest and on community engagement and understanding. SimCoast is also focused on improving modeling techniques and on better understanding the physical characteristics of particles in environmental systems, such as weathering and fragmentation. A huge component of this project is investment in infrastructure, such as improved instrumentation, as well as building a strong foundation of knowledge in microplastics research across the state. And with that, I will wrap up my section and turn things over to Dr. Victoria Fulfer. Thank you very much for your time.
Thank you all so much for being here today. Now that Sarah has given that great introduction into the Narragansett Bay system and what we're finding in the water column, I'll be focusing more on the assessment of plastic pollution on our shorelines and in our coastal ecosystems, including right here in Narragansett Bay, Rhode Island. As we've already talked about, the world has a gigantic plastic problem, and it's been exponentially increasing since the 1950s. We produce nearly 300 million tons of plastic every single year. And what I'm really focused on is the mismanaged part of this waste. So that mismanaged waste is waste that doesn't make it to the landfill or to the recycling plant, but instead somehow, intentionally or unintentionally, it ends up in the environment. So we have over 30 million tons of mismanaged waste just in our coastal areas around the world. And it's estimated that about 8 million of those tons end up in the ocean. But when we measure the amount of plastic in the surface ocean, we're not finding 8 million tons floating around. We're finding tens to hundreds of thousands of tons. So where really is the rest of it? The theory is that most of it resides in the sediment. So we're focused again on microplastics here. And microplastics, as Sarah mentioned, all these small particles, a micron to 5 millimeters in size, that can either be manufactured to be that size or break down to become that small. Just to give you a better idea of what this really looks like in terms of the gigantic size range we're looking at, we are looking down to everything smaller than a red blood cell, all the way up to the size of about the width of an average drinking straw. So a huge size range from things that you can see with the naked eye, such as the ones that we're seeing in this petri dish on the right, to particles that are much smaller where you need a high-powered microscope to see them. So I'll be focusing on two of our most recent studies from a majority of this presentation. The first is an article we published in 2023 showing that microplastics are a problem here in Narragansett Bay sediment. So for this study, we sampled the sediment. Here we're looking at sediment grab samples, which are samples that are taken from the seafloor beneath the water using the sediment grab sampler off the back of the boat. And those are shown in our map here in orange squares. And then we also do shoreline sampling of the sand and the mud of the shoreline, doing 3 transects per area, sampling the upper beach, the middle beach, the intertidal zone, and then going out and doing a subaqueous sample beneath the water as well. And those are shown here on the map as our yellow circles. Just to give an overview of how we look at these microplastics, we look at microplastics down to 63 microns in size. So anything smaller than that, we're not currently looking at. And so 63 to 5 millimeters in size. So we take about 50 grams of sediment and we use a solution of acid to get rid of all of the organic material. So in certain ecosystems, such as marshes and muddy ecosystems, you might have a lot of organics in that sediment, which can interact with your plastic particles. So we want to get rid of all of that. And then we put it in a very dense solution of sodium iodide. This allows the sediment to stay at the bottom while the plastics and less dense material float to the top. And we're able to then separate those two and only look at that low density material, much of which is plastic. So we filter all of that out. And then we're looking at how many particles we're seeing, the color, their size, their morphology, such as if they're a fiber or a foam or a film. And then we're using technology to look at the polymer type. And so we're looking at what type of plastic it is. Is it polystyrene? Is it PVC? Is it polyethylene? Things like that. And to do this, we use micro FTIR or Fourier transform infrared spectroscopy. And basically what this is it uses lasers or light and it shines a laser onto every single particle and it tells us how much light or energy is reflected or absorbed by each particle. So every piece of matter in the world has a different amount of energy or light that it's going to absorb and reflect. And what we get is this squiggly line here, which is a spectra. And this is a little bit like a fingerprint for every type of plastic or really every material on Earth. So if I put a piece of my hair under this, it's going to look differently than if I put a piece of paper under it or something else. And so we match these fingerprints or these different spectral identities to reference libraries, and we figure out, is it plastic or is it not? And if it is plastic, what type of plastic it is. And we do this for hundreds of particles on every single filter. And what we find is, unsurprisingly, that we have a large number of microplastics in the sediment of Narragansett Bay. So here we once more have that map that I showed earlier, just showing where our different sediment samples are. And then in the middle, we have the concentrations of plastic in kilograms per kilogram of dry sediment. And these are oriented just like they are in the map, from north at the top to south at the bottom. So we have Bold Point Park up in Providence, moving south towards Narragansett Beach at the mouth of the bay. And what you can probably immediately see is we have this down system gradient. We have higher concentrations in the north, decreasing as you move to the south. This is seen both in those blue bars, which are shoreline samples, and in those orange bars, which are subaqueous samples. And we're having really high concentrations, over 5,000 microplastic particles in a kilogram of sediment in the north, all the way down to about 10 times lower than that, looking at about 400 particles in a kilogram of sediment when we get down to Narragansett Bay. This makes a lot of sense, of course, because we have much higher populations at the head of the bay. We have the city of Providence, we have other large cities and urban centers, and this allows for just more areas where pollution can happen, but it also means that we have more hardened areas. We have fewer natural areas and more roads and paved areas where stormwater, which often holds a lot of microplastic pollution, is able to flow from these areas into the bay, and it's not being stopped by those grassy or forested areas, of which we do have more of in the southern bay. And overall, what we end up finding when we model all of the data that we have is that about 50% of the plastic mass is being trapped in that upper estuary. It's being trapped north of about Quonset Point. And this is important because when we think about plastic pollution on a global scale, we need to understand what is going from the land to the sea. And in between the land and the sea are these coastal estuarine environments. And so by looking at this, we now know that some of that is being trapped in the estuary. It's not making it out into the open ocean. This, in some ways, is good. Once microplastics are in the open ocean, they're incredibly difficult to clean up or to deal with. But when they're in our coastal environments, we have a little bit better ability to manage them. We also, when we model this, if we were to sum up everything that's in that top 5 centimeters of Narragansett Bay sediment, we're looking at almost 1,000 tons of microplastics just in that top pretty thin layer, which is a pretty alarming amount when you think about the fact that these particles are microscopic in size. So we wanted to understand how we got here. And so we wanted to look at historic plastic pollution levels. And to do this, we used sediment cores. We took 6 sediment cores from beneath the surface of the water, and those are the red dots here. And then we took 3 marsh cores, which are our yellow dots. And cores are a little bit like a time capsule. When you look at sediment underneath the water, it is deposited layer after layer after layer over time. So when you go deeper into the sediment, you're going back in time. So what we can do is take a sediment core and split it into sections and use chemistry to find out around what year that different layer was deposited. And then we can look at how many plastics were deposited during that time range and get a little bit of an idea of how things have changed over time. So what we did is we split all of our cores into 2 centimeter sections. And first we measure lead-210. Lead-210 is a radioisotope. It's naturally occurring in the environment, and it's just a fancy way of being able to tell how long sediment has been on the seabed. And so this is giving us our age, or our sediment accumulation rate. And once we have that accumulation rate, we go from having a depth to being able to have an age. So for example, in this particular site, we were accumulating about 0.3 centimeters of sediment every year. And so then we can convert our depth to a year range. Next, we look at the grain size, or the size of each particle of sediment in each sample. And this is important because the grain size tells us how the energy of the environment has changed. Lower moving calmer water typically means that smaller size fractions, more of that clay is able to be deposited. Higher energy environments with fast moving water, a lot of currents and wave energy, will have higher grain size fractions. And so this just tells us if the environment energy has changed and if it's a lower high energy environment. So we can convert that depth to time. And now here we're seeing the microplastic concentrations through time. And we're seeing them for which year their individual sediment layer was deposited. So we have samples from this particular site down to just after 1950, going to the present day. And you can see that they're increasing over time. Now, this orange triangle you're seeing here exists because when we look at the top 5 centimeters of sediment, remember I said we were splitting each core into 2 centimeter sections. So that top 5 centimeters, which is the mixed layer, this is being impacted by biota, by things that burrow into the sediment, and by currents and wave action. So that top 5 centimeters, that mixed layer, has 2 different samples in it. And so that's just showing the spread between those 2 different samples and the amount of microplastics we're seeing. Next to this, we have the same data, but displayed as a bar graph showing you the different polymer types. So for example, blue here is polyethylene, green is polypropylene, white is polystyrene. And so this is showing how as we increase our concentrations, we are also changing what types of particles we're seeing at different years. So overall, microplastic pollution has been exponentially increasing. This isn't surprising. We've been exponentially increasing the amount of plastic we have been using and creating, and thus putting out into the environment as well worldwide. And so this is reflected in the Narragansett Bay. So here are our top 2 sites. These are the 2 most northern sites, sites 2 and 3. And when we add then our more southerly sites further down in the bay, we see that their concentrations are lower. This is reinforcing that down system gradient that we showed in the previous study. You'll probably also notice that each of these are increasing over time. They have that exponential increase from the 1960s to the present day. Interestingly, when we add our marsh samples, we found something really concerning. So here, microplastic concentrations are ranging anywhere from 500 to 7,000 microplastic particles in a kilogram of sediment, which is about the size of a Nalgene water bottle. If we're looking at those same amount of sediment, but in a marsh, we're looking at 10 to 50 times more microplastics. So now instead of 500 to 5,000 particles, we're looking at 10 to 30,000 particles in a kilogram of sediment. And so our marshes are filtering more of those microplastics and are trapping them in their system. And so when we look at then microplastic accumulation over the past 100 years, we can see how it varies throughout the bay. So these are ordered from north to south, and the ones with the little plant on top are our marsh samples. So you still have that north to south gradient, high to low microplastic accumulation over the past 100 years. But our marsh samples are outliers. They're much higher than the nearest seabed, even down here in the Southern Bay. These two marsh samples are at Fox Hill Marsh, which is on Connecticut Island, which hosts the town of Jamestown. And so this is it's not a very urban area. It's near more rural communities, and it's near the mouth of the bay where you would expect there to be fewer microplastics. However, we're seeing high concentrations in those particular marshes. So overall, we figured out that microplastic accumulation in Narragansett Bay is varying in space, in time, and by environment type. When we redo that model from the first study, but instead look at the last 100 years instead of just the top five centimeters, we can see that about 2,700 tons of microplastics may be accumulating in the sediment. But one of my favorite outcomes of this study was this plot right here. We were able to see, based on the large number of samples that we took, microplastics from 1940 to 2020, and we see how their concentrations are increasing throughout that time. We're also seeing how the diversity of polymers, of plastic types, has increased over time. So we start in times where only a few plastic polymers even existed in the world, and then we get to the present day where there are hundreds of varieties of different plastics and dozens of classes. And so we can really see that pattern showing up in the sediment record. So overall, we're finding that Narragansett Bay sediments have exponentially increasing microplastics, with sites that are closer to urban centers being more highly polluted than sites further away. Marshes are trapping 10 to 50 times more microplastics than the nearby seabed. And the diversity, whether it's the morphology, the color, or the polymer type, has been increasing over time, which really complicates pollution issues because it means that all of these have different chemical profiles and will behave differently, whether it's in the environment or in the body. But what really are the consequences of this? Unfortunately, when it comes to ecotoxicology data, you really need species-specific data, and we don't have a lot of that yet for Rhode Island. But we do have colleagues who are beginning to work on this. But other studies have been done in similar types of estuaries, and I'm showing one of those here. And this estuary had similar organisms to what we find here in Narragansett Bay. And when they completed their ecotoxicology study, they found that the threshold for harm was about 540 microplastic particles in a kilogram of sediment. So anything above that means that more than 10% of the population is going to be impacted. And anything below that means that less than 10% of the biological population is going to be impacted. And so let's see what would happen if we apply that to Narragansett Bay. So on the left, we have all of the different samples that we took throughout all of our studies. They're colored and they're shaped based on what type of sample they are. I have highlighted here in pink the waters that are impacted by pollutants transported with stormwater runoff according to the DEM. And so we could expect that these will have more highly polluted sediments and waters in this particular region. And then to the right, it's those same samples, but now they are colored by how far above or below that risk threshold they are. So anything that is green is below the risk threshold, and then yellow, orange, and red are above the risk threshold. And unfortunately, you're probably seeing a lot more yellow, orange, and red than we are green. So shorelines and the seabed in the proximal zone are well above that risk threshold. In the distal zone, this lower part of the bay, we do have some that exceed that level, but many of the others are still below that risk threshold. So while we still need more data specific to Narragansett Bay organisms before we can really know what the negative impacts might be, this preliminary look is pretty concerning when we think about the number of sediment, the number of organisms that reside in the sediment. We're thinking about our shellfish, we have bottom feeding organisms, things like that. And all of this really comes together along with much of my other work and makes me think about how the way we structure our coastal systems and our coastal communities can really impact the propagation of pollution signals within a system and the export of pollution outside of the estuary. So some estuaries are natural estuaries. We don't have populations living in them, and these are pretty rare around the world now. But when you have a natural estuary, you have marsh shorelines, sandy shorelines that are able to trap some of that microplastic. So it's not really getting out into the environment. As we start to build in our estuaries, we're starting to notice more input of those pollutants, and also a decrease in the types of environments that can capture them. This is called hardening of the shoreline. So hardening of the shoreline is when you build a seawall, you add riprap, you get rid of sandy or marshy shorelines and instead create more developed areas. That's hardening of the shoreline. And this hardened shoreline cannot trap microplastics efficiently. And so it's starting to create a funnel to the open ocean. And unfortunately, many of our estuaries in the world look like this. They're highly urbanized. They have a lot of hardened shorelines and very few environments left that are able to trap microplastics. This is why it is so important to maintain our marsh environments and, when possible, to allow the development of new marsh environments so that we can filter our pollutants before they make it into the open ocean. So overall, increasing that hardening and urbanization of our coastal zones is increasing sources of plastic pollution and offshore export. And we can see this when we look at other data. I completed a study with the ocean race in 2023, where the sailors raced all around the world and collected microplastic samples for me as they went. So you can see their route here, and the larger and more darker circles have more microplastic pollution than the smaller and lighter colored circles. And so we can see here where there are certain hotspots, particularly near coastal zones. But what was really concerning is that even in these areas very far from land, including this dot here, which is near Point Nemo, where the closest person to you when you're at that point in the ocean is in the International Space Station up in space. So we're incredibly far from land, but we're still finding microplastics in those areas. So we know that there is still export of microplastics from coastal areas to the open ocean. And by changing the way that we develop our coastal systems and the way that we manage our marshes and sandy ecosystems, we're able to hopefully filter more of those pollutants. And this is where my new work with Gyres Institute comes in. Gyres is a solution-based, science-based nonprofit where we're trying to look at many different solutions to the plastic pollution problem. And one of those is nature-based solutions. And so you'll remember we talked about how the marsh environments are accumulating all of these microplastics, but we don't have a huge number of marsh environments in many of our coastal ecosystems. They're constantly being either developed over or are being impacted by sea level rise and are unable to keep up. And so we wanted to take the idea of marshes working as a filter for microplastics and see if we could implement this in an area where there isn't currently marsh ecosystems by using floating wetlands. So floating wetlands create a wetland ecosystem on a float that is out in the water and acts very much like a coastal wetland without needing the actual remaining land. And so floating wetlands have been used a lot, particularly in the southern United States in retention ponds to absorb nutrient pollution, and has also been used to take up pollutants from urban runoff and pesticides. And so we know they work to filter certain pollutants, and we wanted to see if they also could work to filter microplastics. So we built floating wetlands this past year and deployed them throughout the state of Rhode Island. The one pictured here is at the Providence Save the Bay Center. And we wanted to make floating wetland systems that were completely plastic free. This is a difficult thing to do. Often when floating wetlands are built for retention ponds, the float bottom is actually made of plastic because it's lightweight and it floats. But since our goal is to remove plastic from the environment, not add it, we instead use micro rafts. Micro rafts are an amazing invention by Sue Van Hook. And she uses heat-treated mycelium, and mycelium is a fungus. These are naturally occurring fungi. And so they're heat-treated, so they're no longer growing. And it creates a float that is biodegradable and perfectly safe and natural for the environment and will degrade over time. And then we top that with a coconut coir mesh, which allows it to have structure and allows something for the plants to hold onto. And then because we're here in Rhode Island, we use Spartina alterniflora plants. These are the most common marsh plants here in the state. And so we deployed these starting in the spring and went all the way through until October. And what we're looking at is we took a time series of the amount of pollution in the water, any microplastics larger than 20 microns, over the course of every other week in the surface water to see how much microplastic pollution was at each site. And then we are sampling the floating wetlands themselves to see how much microplastic pollution they are trapping within their plant matter and within the raft. And then we're also looking at the amount of pollution in the sediment in the surrounding ecosystem. So that is still ongoing, but I wanted to talk a little bit more about some of our other work that we have ongoing as well. So we talk a lot about microplastics in the ocean and the water, but they're not just in the water or in the sediment. They are also in the air. There's a huge, every day as you're breathing, you are breathing in microplastics, unfortunately. Microplastics, one of the most common airborne types are microfibers. Microfibers come off of our laundry or when we're wearing our clothes, from our carpets, things like this. And they can be spread, whether it's through urban centers or from the solid waste application from wastewater treatment plants to agriculture. But other sources include road dust and sea spray, so the microplastics in the sea spray coming off into the air. And so we wanted to look at source reduction of microfibers from textiles. Microfibers, as I mentioned, are small threads smaller than 5 millimeters that shed from textiles, and they can be synthetic or plastic or natural. So they can be something like polyester, or they could be something like cotton. And textiles are a pretty understudied portion of the plastic pollution problem, but they actually make up 10% of plastic production every year. And textiles are the second most discarded type of plastic in the world, with Americans generating almost over 16 million tons of textile waste every year. Every time you wash your clothes, you're releasing about 18 million microfibers into the wastewater treatment stream. Many wastewater treatment plants are able to remove many of these fibers, so they're not getting into our waters. However, we often also use our wastewater treatment sludge as fertilizer on agricultural systems, which puts those microfibers back into the environment either way. So when we launder our clothes, washing and drying, everyday wear, and when the use of home textiles, we're adding microfibers to the environment. And one of our recent studies that came out this month was that a single laundromat can release over 7 trillion microfibers into the air each year, which is a huge amount when we think about the number of laundromats, particularly in our cities around the world. So we wanted to understand it. We know that washers are releasing microfibers, and we know that dryers are releasing microfibers. There's a lot of work done on secondary filtration for washers, but less done on secondary filtration for dryers. So we wanted to test three options for secondary filtration that would capture microfibers that make it past your lint filter. So often when you have a dryer in the United States, you will have a vent that vents air, hot air, out to the outside of your home. And you might have noticed that you have some lint and microfibers accumulating in this area. And that's because millions of microfibers make it past your lint trap and out into the air. So the three fibers that we tested, one was a duct filter that goes directly onto your dryer duct, and two others were ventless indoor filters, which stops air from being vented outdoors at all from your dryer. What we found was compared to the control, here in purple, our duct filter and our other two filters reduced the amount of pollution, but our indoor filters reduced it the most. And we also, that you can see this both in the amount of microfiber mass and the count of microfibers trapped by each. And this is a little bit of an ideal solution. It's easy to install, it's cost effective, and many states could lobby for having them installed on newly manufactured machines, similar to bills that have already been introduced for washing machines in California, Oregon, New Jersey, and New York. But overall, the most important thing is to do less. Buy less clothes, wash your clothes less often, and be less wasteful when it comes to your textiles. One thing that we spend a lot of time looking at is how we can work with industry so that the burden isn't on the consumer, it's more on the manufacturer. And so we created a microfiber playbook. And this is just one snippet of this playbook, which is looking at how different designs of textiles and manufacturing methods impact microfiber shedding. So the way that you weave fabric, the dyes that you use, the way that it is cut, all of this impacts how much a shirt or a jacket or whatever it is going to shed. And so we got together with industry and the leaders in the textile world, and we created this chart that shows which ones increase shedding and which ones decrease shedding so that they can make more informed choices. And that brings us to advocacy. This is a huge part of Five Gyres. We have an amazing policy team that advocates both the state level in various states as well as at the national level in DC. We led the Ban the Bead campaign, which led to the passing of the Microbeads Free Waters Act in 2015, and have also supported the creation of the Fighting Fibers Act of 2024 and the ban on plastics in US national parks. That has unfortunately now been rescinded due to the current administration, but we are hoping that we will be able to continue this fight and get some of these re-implemented with future administrations. But what's something that you can do? One thing that we created was an app called Trash Blitz. This is an app that's free on whatever kind of phone you might own, and it allows you to be part of a community science-based project. So when you're walking your dog or taking a walk on the beach or really anywhere, and you see trash, you can put it into the app. We tally both the types of items that you're finding, what they're made out of, whether it's plastic or paper or metal, and also what brands they are. So we care a lot about brands because we want to be able to hold brands accountable. And so whenever it's possible, we like to know who created the product that has been littered into the environment. And this ends up giving us data from all over the world where we're able to see the top items being littered, typically cigarette butts, followed by wrappers and bottle caps, and the top brands, Coca-Cola, Marlboro, McDonald's, all the ones that you might be able to expect. And this data is really important, and we create reports every year that help policy members and community members advocate for change when it comes to litter and management of waste. So overall, if you want to reduce plastic pollution in your own life, the biggest thing you can do is whatever you're going to be able to do consistently. So if that means reducing single-use plastics and using your reusable coffee mug, that's great. If that means picking up trash on your daily walk or going to a thrift store instead of buying new clothes, that's also great. Whatever you can do will make a difference because if we all do it together, that collective action can have a huge impact. And I highly recommend taking advocacy action if there's any pollution related policies coming up, whether at the state or local level or nationally. Overall, I just want to thank all of our funders and collaborators for all of these projects, and I would be very happy to take any of your questions. Thank you.
And I see an earlier question about effective capital infrastructure investments to reduce the transport of microplastics from the environment, asking about trash skimmers or river mouth booms, things like this. One thing I always like to say is any solution is better than no solution. You know, we don't have a perfect solution right now for plastic reduction. None of these solutions are going to reduce or are going to trap microplastics. So trash reels, trash skimmers, the booms, those are collecting macroplastics. So it will reduce the amount of microplastics in the environment, you know, down the line because those are being removed from the environment, will not break down into microplastics. But we don't currently have a way of just filtering all of the microplastics out of the water or removing them from the sediment. Adjusting our wastewater and stormwater systems, in my opinion, are really great ways to reduce the amount of microplastics making their ways into our waterways. Stormwater is a huge area of emissions of microplastics into the bay and into all kinds of environments. And so improving our stormwater systems would be one really great way. And probably, you know, it's obviously it's costly, but if we're really focused on reducing microplastics in particular, that would be the way that I would go. I don't know, Sarah, if you have any other opinions on that.
No, I definitely agree. I think all potential solutions have trade-offs, and what works in one neighborhood might not work in another. But the best thing that we can do is gather as much data as possible before implementing those technologies to be best informed about what types of microplastic pollution, what possible sources, and that can help us identify like what solutions might be most effective.
And I will just add, I haven't had a chance to look over everyone who's on the call, but we are putting together a working group for nature-based solutions and also other types of infrastructure solutions for microplastics. And so if anyone is working in that space and would be interested in being involved, please reach out to me because we are trying to get a large group across the nation together to really think about these solutions and work together to try to pilot some of them.
I did see Meredith briefly reappear. Meredith, are you back?
Yes, kind of. Sorry for that, everyone. Um, yeah, where are we?
We've, I think, gone through a lot of the chat questions.
Okay, great.
I think maybe most of them. It's sort of hard to keep track. Victoria, did you see any outstanding ones that we had missed?
I
Or maybe,
think we got
I
most
think
of
most
them.
topics have generally been covered.
Okay, great. Again, sorry for my technological difficulties on my end, but I'm glad everyone was able to carry on and work through the questions. There were some really good, great questions I remember seeing in the Q&A. Was there anything else that anyone wanted to ask? It's like,
Ooh, big question about,
ooh.
um, interesting question about microplastics in human bodies. From an interesting perspective, I think this idea of um, maybe folks donating their body to science so we can identify microplastics at end of life, which is really interesting. I don't know of any studies that are doing that specifically. There are certainly a number of studies now published that have identified microplastics in a range of human tissues. Those can be really startling studies. I will say, I think it's very important to really critically read the methods of every study, especially those that are identifying plastics in human tissues, to identify their contamination controls, um, especially when we think about how human tissues are stored in like these tissue banks and how they might have originally been recovered and processed and then stored. A lot of times that was not done with microplastics analysis in mind as the end goal. I just want to put that out as a caveat. I'm not saying that these are not accurate studies, but I found it challenging sometimes to reconcile some of the methods of what they're finding with what their potential methods are. That's not a direct answer to the question, but it's just my plug. I don't know if anyone else, if Victoria, you want to jump in and talk a little bit about that human health aspect.
Yeah, in terms of the question itself, I don't know of any efforts in particular partnering with those services. Yeah, I don't know of any, but I do agree with Sarah that, there's a lot of human health studies coming out right now. And it definitely, one of the biggest issues with microplastic science, regardless of where you're studying it, whether it's in the body or in the environment, is a harmonization of methods. And so we don't, because it's such a new field, there isn't necessarily that really good harmonization of methodologies yet. And so it can be difficult not only to validate studies, but also to compare between studies. And so this is becoming especially important as we're looking into human health, where we're looking at nano-sized plastics, even smaller than the microplastics. And so definitely concerning that we're seeing more and more of these studies come out, finding them in human tissues. But I think we're a long way off from really knowing what the impact on human health is. I think we're going to need a lot more research before we kind of get to that point.
Yeah, absolutely. I agree. And I think there's been some really cool research coming out of the University of Rhode Island about the impact of microplastics on brain health by some folks in neuroscience, which is really cool and interesting and necessary work. And we're excited to see that continue. And as Victoria mentioned, this idea of methods harmonization has been an issue for microplastics research since it kind of was coined. Like the first study naming or coining the term microplastic was, I think, in 2004. So it's a new field and it kicked off very quickly and people started studying things in different ways. So it was challenging to compare results. We now have really robust best practices for how to study microplastics, but there are some lingering issues that have persisted, or some maybe calls for additional harmonization as we move forward. And one of those big things is when we do research on the effects of microplastics, and we're taking organisms, say, for example, marine organisms, and exposing them to a known quantity and type of microplastic, that those types and amounts are realistic. Early microplastics research tended to use doses and types of microplastics that are not only unrealistic in the environment, but also in terms of their dose, like they were much too high of a dose, but they also were not representative of the actual types of plastic you would find. So they tended to use microbeads, for example. We don't really find microbeads in the environment. They're very scarce. We tend to find more secondary microplastics like fragments and fibers. And so more recently, these exposure studies looking for effects have used much more realistic quantities and types of plastics in their exposure work so that when we see a potential effect, whether that's something like growth impairment or issues with reproduction, that we have a better sense of like what that threshold really looks like, and that we can contextualize it based on the real pollution scenarios in the environment.
Yeah, a question about how microplastics interfere with the metabolism of organisms in the sea. And I kind of wanted to tag on, it might be a separate question, but it might be related, and you might have already answered it, so forgive me, about the types, you mentioned, Victoria, what were common in the 1950s and some of these more modern types like PET that are found, and what kind of different fragmentation rates they would have and how that might impact the metabolism rates.
Really interesting question. I'll kind of start with your question, Meredith. Um, in terms of fragmentation rates, that's a really difficult question to answer because all of these different types of polymers can be made into so many different types of plastic products. And these products will have different flexibilities, they will have different thicknesses, different uses. And so their fragmentation tends to differ depending on how they were created. And this is one of the reasons why studying microplastics and predicting from source material to microplastic pollution, kind of like what those sources might be, because, all of these plastic products not only have that, kind of parent polymer type, whether that's PET or something else, but also all of these additives that change their properties to make them more flexible or heat resistant or whatever it might be. And then more chemicals in order to, change their durability and change their color and things like that. And so it's really complicated in terms of fragmentation. But I think this relates to the interference with the metabolism of organisms because, it's not just the plastic that is being ingested, it's all of those chemicals and all of those additives. And then we're also seeing more and more information coming out about the co-location of microplastics with other types of contaminants, such as pesticides, chemical contaminants, heavy metals, and how some of these can adsorb to the plastic particles themselves and then make it into the organism's bodies. And so it becomes incredibly complicated to study the impact of microplastics on an organism because you have to try to differentiate between, is it the plastic particle causing this? Is it a chemical that was added to it? Is it another thing that was adsorbed onto it from the environment? So it becomes very, very complicated. But in answer to Nick's question about how good the evidence is, today we have fairly strong evidence for many organisms, not for every organism in the sea, but for many organisms. In some ways, many sea organisms that we've used in these ecotoxicology studies have shorter lifespans than a human, so they're much easier to study the impact. Humans obviously live a very long time, we grow very slowly, so it's a little bit harder to study human health in terms of if, we're finding, there are studies coming out that there are microplastics in newborn babies, with these microplastics being passed from the mother. If this is the case, that means, throughout your entire life, you have microplastics in you and you might be accumulating more. And we're trying to figure out what that might mean. It's really hard to study in humans when we've only known about microplastics for about 20 years, when the human lifespan is obviously much higher than that. But in a marine organism that grows much more quickly and reproduces much more often, it's a little bit easier to understand some of these impacts. And so these days we have a lot of more high quality studies on many marine organisms, and we are seeing those negative impacts to metabolism, to their hormones, to DNA replication, stress, all of those things. And so definitely still work needs to be done, particularly on larger organisms in the ocean. But we do have some pretty good evidence now that we can say that there is an impact. It's just sort of a matter of how far reaching is that impact going to be.
Absolutely.
And
And oh, go ahead, Meredith.
no, because I was going to ask another question, but I want you to follow up on that one if you have other thoughts.
Yeah, I was just gonna note that I shared a resource in the chat. It's called the TOMEX, or the Toxicity of Microplastics Explorer, and this is a resource that was put together by a group called oh, this name is long, Southern California Coastal Watershed Research Project, or SCWRP for short. And they've synthesized a ton of studies that have done exposure work, with aquatic organisms, and it's interactive, so you can search by organism or different variables of the study and see what they found. And it's great too because they use some evaluation criteria that I think are helpful to read about to better understand what maybe these kind of evaluators are looking for when people are putting together an exposure study. Interesting question about microplastics in human bodies. From an interesting perspective, I think this idea of maybe folks donating their body to science so we can identify microplastics at end of life, which is really
Oh,
interesting. I don't know of any studies that are doing that specifically. There are certainly a number of studies now published that have identified microplastics in a range of human tissues. Those can be really startling studies. I will say, I think it's very important to really critically read the methods of every study, especially those that are identifying plastics in human tissues to identify their contamination controls, um, especially when we think about how human tissues are stored in like these tissue banks and how they might have originally been recovered and processed and then stored. A lot of times that was not done with microplastics analysis in mind as the end goal. I just want to put that out as a caveat. I'm not saying that these are not accurate studies, but I found it challenging sometimes to reconcile some of the methods of what they're finding with what their potential methods are. That's not a direct answer to the question, but it's just my plug. I don't know if anyone else, if Victoria, you want to jump in and talk a little bit about that human health aspect.
yeah, in terms of the question itself, I don't know of any efforts in particular partnering with those services. Yeah, I don't know of any, but I do agree with Sarah that, there's a lot of human health studies coming out right now. And it definitely, one of the biggest issues with microplastic science, regardless of where you're studying it, whether it's in the body or in the environment, is a harmonization of methods. And so we don't, because it's such a new field, there isn't necessarily that really good harmonization of methodologies yet. And so it can be difficult not only to validate studies, but also to compare between studies. And so this is becoming especially important as we're looking into human health, where we're looking at nano-sized plastics, even smaller than the microplastics. And so definitely concerning that we're seeing more and more of these studies come out, finding them in human tissues. But I think we're a long way off from really knowing what the impact on human health is. I think we're going to need a lot more research before we kind of get to that point.
Yeah, absolutely. I agree. And I think there's been some really cool research coming out of the University of Rhode Island about the impact of microplastics on brain health by some folks in neuroscience, which is really cool and interesting and necessary work. And we're excited to see that continue. And as Victoria mentioned, this idea of methods harmonization has been an issue for microplastics research since it kind of was coined. Like the first study naming or coining the term microplastic was, I think, in 2004. So it's a new field and it kicked off very quickly and people started studying things in different ways. So it was challenging to compare results. We now have really robust best practices for how to study microplastics, but there are some lingering issues that have persisted, or some maybe calls for additional harmonization as we move forward. And one of those big things is when we do research on the effects of microplastics, and we're taking organisms, say, for example, marine organisms, and exposing them to a known quantity and type of microplastic, that those types and amounts are realistic. Early microplastics research tended to use doses and types of microplastics that are not only unrealistic in the environment, but also in terms of their dose, like they were much too high of a dose, but they also were not representative of the actual types of plastic you would find. So they tended to use microbeads, for example. We don't really find microbeads in the environment. They're very scarce. We tend to find more secondary microplastics like fragments and fibers. And so more recently, these exposure studies looking for effects have used much more realistic quantities and types of plastics in their exposure work so that when we see a potential effect, whether that's something like growth impairment or issues with reproduction, that we have a better sense of like what that threshold really looks like, and that we can contextualize it based on the real pollution scenarios in the environment.
See a question about how microplastics interfere with the metabolism of organisms in the sea. And I kind of wanted to tag on, it might be a separate question, but it might be related, and you might have already answered it, so forgive me, about the types, you mentioned, Victoria, what were common in the 1950s and some of these more modern types like PET that are found, and what kind of different fragmentation rates they would have and how that might impact the metabolism rates.
Really interesting question. I'll kind of start with your question, Meredith. Um, in terms of fragmentation rates, that's a really difficult question to answer because all of these different types of polymers can be made into so many different types of plastic products, and these products will have different flexibilities. They will have different thicknesses, different uses, and so their fragmentation tends to differ depending on how they were created. And this is one of the reasons why studying microplastics and predicting from source material to microplastic pollution, kind of like what those sources might be, because, all of these plastic products not only have that, kind of parent polymer type, whether that's PET or something else, but also all of these additives that change their properties to make them more flexible or heat resistant or whatever it might be. And then more chemicals in order to, change their durability and change their color and things like that. And so it's really complicated in terms of fragmentation. But I think this relates to the interference with the metabolism of organisms because, it's not just the plastic that is being ingested, it's all of those chemicals and all of those additives. And then we're also seeing more and more information coming out about the co-location of microplastics with other types of contaminants, such as pesticides, chemical contaminants, heavy metals, and how some of these can adsorb to the plastic particles themselves and then make it into the organism's bodies. And so it becomes incredibly complicated to study the impact of microplastics on an organism because you have to try to differentiate between, is it the plastic particle causing this? Is it a chemical that was added to it? Is it another thing that was adsorbed onto it from the environment? So it becomes very, very complicated. But in answer to Nick's question about how good the evidence is, today we have fairly strong evidence for many organisms, not for every organism in the sea, but for many organisms. In some ways, many sea organisms that we've used in these ecotoxicology studies have shorter lifespans than a human, so they're much easier to study the impact. Humans obviously live a very long time, we grow very slowly, so it's a little bit harder to study human health in terms of if, we're finding, there are studies coming out that there are microplastics in newborn babies, with these microplastics being passed from the mother. If this is the case, that means, throughout your entire life, you have microplastics in you and you might be accumulating more. And we're trying to figure out what that might mean. That's really hard to study in humans when we've only known about microplastics for about 20 years, when the human lifespan is obviously much higher than that. But in a marine organism that grows much more quickly and reproduces much more often, it's a little bit easier to understand some of these impacts. And so these days we have a lot of more high quality studies on many marine organisms, and we are seeing those negative impacts to metabolism, to their hormones, to DNA replication, stress, all of those things. And so definitely still work needs to be done, particularly on larger organisms in the ocean. But we do have some pretty good evidence now that we can say that there is an impact. It's just sort of a matter of how far reaching is the impact going to be.
Absolutely.
And
And oh, go ahead, Meredith.
no, because I was going to ask another question, but I want you to follow up on that one if you have other thoughts.
Yeah, I was just gonna note that I shared a resource in the chat. It's called the TOMEX, or the Toxicity of Microplastics Explorer, and this is a resource that was put together by a group called the, oh, this name is long, Southern California Coastal Watershed Research Project, or SCWRP for short. And they've synthesized a ton of studies that have done exposure work with aquatic organisms, and it's interactive, so you can search by organism or different variables of the study and see what they found. And it's great too because they use some evaluation criteria that I think are helpful to read about to better understand what maybe these kind of evaluators are looking for when people are putting together an exposure study.
Thank you for that resource. I think the only other questions I had, and again, you might have already covered this, was, as they break down and, they have these other chemicals and additives, but I've also read somewhere about how microplastics can be vectors for other marine diseases as they pass around. And I guess that's kind of more surface related. But then did you guys discuss kind of the impacts of like warming waters, increased storm intensity on whether that has, I guess, the impacts that we're seeing with climate changes, how that might impact distribution, fragmentation rates, and I guess maybe this now kind of vector component, aside from transporting other chemicals and additives, but other just marine diseases.
It's a big question. Um, I can jump in and say there's definitely connections between climate change, the effects of climate change, and microplastic inputs and distributions. Um, it's challenging to say precisely what relationships are going to be affected and how. We know that there tends to be increased microplastic suspension after major storm events. That's something that has been demonstrated pretty consistently in the literature. A lot of that just has to do with the bioturbation that's happening. It's kicking up a bunch of sediments. It's, washing a ton of stuff off of the land into the nearby waterways. With warming actions and possible like changes to ocean dynamics and currents, that's something that's kind of been preliminarily hypothesized in terms of microplastics as vectors. We know that different communities tend to colonize microplastics, like microbial communities, than natural materials that are in the same waterway. That's also been well demonstrated. But the question mark right now is we don't know what that looks like in the long term, or across these like vast distances. But I think there's definitely the potential that's there, and that is an active area of research is what does this mean? Also, as we see warming happening and like the dynamics of all of these ecosystems are changing and what can survive in some areas is changing. Um, yeah, Victoria, if you want to jump in.
No, I think you really answered that super well. And I do think that with this warming and this changing chemistry of the ocean, we are going to see, the dynamics of transport and fate also change, unfortunately, with microplastics, which is I say unfortunately because we don't even have a good handle quite yet on how it works now, and now it's in a changing environment that makes it even more difficult. And in terms of the vector of those diseases, that's definitely a huge concern. It's a pretty prolific area of research right now where they're trying to understand that. I think we will see that kind of expand with this warming, unfortunately, as well. But we don't have all of the information yet.
Thank you so much. I want to leave it open for a quick minute. Does anyone else have any other questions that they would like to ask? Or to you, Victoria or Sarah, if there's anything else that you wanted to add that you might not have covered. I feel
I
like you
think
guys
we've
covered great
covered a
things.
lot.
Yeah, yeah.
No, this has been really wonderful. It's been great to learn more about Victoria's work, especially with five gyres, and think about how we can translate all of this research into advocacy on the policy side, especially for our state of Rhode Island and what that can look like. And I really appreciate everyone joining in the chat, especially and contributing to those conversations.
Absolutely.
Well,
We got some really amazing questions. So thank you, everybody.